Today we'll finish with conversation about simple Mendelian genetics, and in particular we'll talk about sex-linked genetics and pedigrees. So let's start with sex determination. We'll see how there is sex determination on the one hand, and then what we mean by sex-linked genetics. We'll discuss that in a second. They go hand in hand. In humans, as most likely all of you know, the sex determination system is given by the sex chromosomes, the XX, XY system. The way this works is individuals who have two X chromosomes will usually, and I stress usually because of a discussion that we'll have in the next couple slides, they will usually have female reproductive organs and make female gametes. And then XY individuals will have male reproductive organs and gametes. And this applies to all mammals, right? We'll see how other types of animals have different sex determination systems, and we'll describe those briefly. So in mammals, the XX-XY system works such, this some of you may not know, works as follows. The reason why individuals with a Y chromosome develop male reproductive organs and make male gametes is because the Y chromosome carries a gene called the SRY gene that whose protein product functions very early during embryonic development. And the way that this works is when the SRY gene is present and expressed in XY embryos, its protein product will target embryonic gonads that are up to that point neutral. So the embryonic gonad is developing without developing any female or male characteristics. But then if the SRY protein is present, the embryonic gonad will become an embryonic testis. It will start expressing male sexual hormones, which will in turn trigger the development of a series of secondary male traits, including the formation of those male genitalia and the later on the production of male gametes of sperm. In XX embryos, if the SRY gene is not present and therefore not expressed, the embryonic gonad defaults into an embryonic ovary. It will start expressing female hormones, sexual hormones, which will in turn trigger the development of secondary female traits. Now, the reason I was emphasizing before that the presence of an XY is usually associated with male anatomical characteristics and XX usually associated with female anatomical characteristics is because that's what's happened usually, but there are exceptions to this. So a question that I have for all of you, let's say that there is an XY individual. Can you imagine a scenario in which that XY individual may not develop male anatomy and gametogenesis? I heard something. What? Yes. Can you imagine it? What would that scenario be?
I don't know if I'm right, but it reminds me of Klinefelter syndrome.
What was Klinefelter syndrome? Remember?
They have the XXY,
Uh-huh.
an extra X or something like that.
Uh-huh. But were those? Did those individuals have male anatomies?
Female parts.
They had female parts.
I think.
No, but you're thinking on the right way. So an XY individual, how could an XY individual not express male anatomy and gametogenesis based on what we just described? When could an XY individual not develop secondary male traits? You mentioned the XY. What other type of Turners? What was Turner? Remember?
It was missing.
A missing one, right? So how could an XY individual, for all intents and purposes, be like an X? No.
Blank.
X blank, yeah. What could happen to that Y chromosome such that now all of a sudden it's not behaving like a Y chromosome anymore? Of one. Yeah. Of one. Of one. There's the SRY gene, exactly. Let's imagine, and this happens. Let's imagine an individual is, their karyotype is XY, but the SRY gene is mutant. Is not there. Deleted. For all intents and purposes, deleted. That individual has a Y chromosome, but they will not develop male characteristics because they're missing the SRY gene that expresses the SRY protein that triggers a cascade of developmental events that will lead to the formation of secondary male traits. Right? So as a mutation in the SRY gene is possibly the most obvious scenario in which an XY karyotype will not lead to male anatomy in humans or mammals in general. But that's not the only one. There's a series of events that take place here, all of which depend on the production of certain hormones, the reception of those hormones, the response to those hormones. Each of those are controlled genetically. And therefore, there could be a series of variations or mutations or genetic modifications to those steps in the cascade that sort of unlink the presence of a particular karyotype, in this case XY, from developing those secondary male traits. Okay? This also opens the door for a discussion that has become has come to the surface lately, and that has become a little politicized recently, and that is, what is the biological basis of gender and sex? You guys probably heard that even politicians now, congressional hearings and whatnot, they're going to be throwing around, what is a woman, kind of question. Have you guys faced that question from friends or relatives? When you say, yeah, I'm a bio major, did they immediately hit you with a question, by any chance? Some of you? Some? Do you have an answer for it? It's a tricky one. It's a complex topic. So hopefully I'm going to give you some elements to think about this question from a biological perspective a little more clearly. So let's extend that argument. We mentioned here secondary male traits. And it is fair to say that among those secondary male traits, some of them are visually more obvious than others. So anatomy, gametogenesis, the ability to make sperm versus eggs, right? Some of them may be a little less obvious, like things related to behavior, or one very specific case of, or very specific kind of behavior, which is self-perception and identity. Right? So one might imagine that certain aspects of gender identity are, if not secondary, tertiary, quaternary, some level of indirect effects of the cascade of events that lead to these type of dimorphism between XY individuals and XX individuals. When you think about it that way, that kind of lends some credence to the argument that identity is biological. If you're XY, you should be a cisgender male. If you're XX, you should be cisgender female. It's biology. But we just made the argument that there may be certain genetic variations that happen along the way that change things. So that argument can be easily applicable to aspects of identification and gender identity. If we perceive gender identity as one of very diverse range of psychological traits, right? I don't see why they couldn't have a biological basis. So this whole idea that, oh, you're XY, so you should identify this way or another, is biologically speaking a moot argument, because we don't know how many steps link karyotype to gender identity, if that makes sense, right? So if you ever face the question, the official biology answer is that anatomical anatomy, or sexual anatomy and gametogenesis, the ability to make eggs or sperm, are separate phenotypes from things that have to do with self-perception and identity. And both are equally biological, right? So this idea of, someone who's not identifying with their sex at birth are making a choice, or all of that is moot. There can be a biological basis that is not necessarily dictated by the presence or absence of a chromosome. Did that make sense? Did this trigger any questions in anyone that you feel comfortable sharing? It's a thorny aspect. A few years ago, we wouldn't really spend any time talking about any of these, but it has become a thing. And as bio majors, you guys need to be equipped to handle this question out there because you're going to get asked. No? Okay. So let's move on. I said before that there is different sex determination systems in different. This slide is about animals because in plants there is a lot of mostly hermaphrodites, though there are some plants that have male and female individuals. But this slide is about sex determination systems in animals. So we already mentioned the XX, XY system in mammals. There is the X to autosome system that is present in two very famous model organisms in research, and of course many other invertebrates. In this system, XX is female and XY is male. You'd be inclined to think, oh, just like in mammals. But the difference is that in those organisms, XYs are male, not because the Y chromosome carries a determinant like an SRY gene that will trigger a series of dimorphism cascades, but because what makes those individuals male is a halved ratio of X chromosomes to autosomes. And that became apparent when researchers noticed that X null individuals were not female as you would expect due to the absence of a male Y chromosome, but were instead male because what they had was a half ratio of X chromosomes to sex chromosomes. So in these organisms, what matters is what is the relative proportion of X chromosomes to the rest of the chromosomes in the animal. And an X0 is like an XY because in both cases you have only one X chromosome relative to all of the other autosomes in the organism. Did that make sense? Cool. Birds, snakes, butterflies, amphibians, fishes have the ZZ-ZW system, similar to the XX, XY system, except that the what is called the homogametic sex is male, meaning that when individuals have two of the same chromosome, in this case the Z chromosome, they're male, and the one with the heterogametic individuals are female, right? And we call them homogametic or heterogametic because they will be producing only one kind of gamete, the males, or the two kinds of gametes, the females. When you think about it, in humans, it's the other way around. Males produce two kinds of gametes, right? Sperm with the X chromosome or sperm with the Y chromosome, whereas females always produce X with the X chromosome, because that's the only type of sex chromosome they have.
For this system for butterflies, is it caterpillars and butterflies, or is it just butterflies?
Butterflies are grown-up caterpillars. They're the same.
Well, I was just wondering during the cocoon phase, it develops a system.
Oh, the expression? Yeah. Well, I don't know enough about butterfly biology. In flies, for instance, in fruit flies, you also have a larval stage, the equivalent of the caterpillar, and there is already sexual dimorphism at the larval stage. So I would imagine that butterfly caterpillars also have some sort of sexual dimorphism, but I don't know the details.
So you can tell by just a larval stage.
If you know enough, yes, yeah. If you know enough about caterpillars and larvae, you can look at them and tell. Like you give me a fruit fly larva, and I can tell you if it's a male or a female. All right. Then there is the haploid-diploidy system. Haploid-diploidy system, similar in a way to the X to A system, in that the individuals with only haploid set of chromosomes are male, and individuals with a diploid set are female, and this happens in bees, wasps, and ants. I often get the question, oh, those seem to be social animals, right? Bees and ants mostly. Does that have to do anything with the being a social species? I don't know. It could be coincidence too. I personally don't know. Now, in all these cases, the details differ across animal groups, but in all these cases, the underlying biology is generally the same. And that is that you have a series of genetic events that happen in a cascade that will lead to some sort of trigger that may have to do with the presence of a given gene, like in mammals, or a certain ratio of X chromosomes to autosomes, or the presence of the W chromosome in bird, snakes, and so forth. There is some sort of initial trigger that will initiate a series of genetic events that lead to the development of anatomies and gametogenesis, or the ability to make one type of gamete or the other. But these are genes, right? So there is even examples in which, like any other phenotype, there is a strong environmental influence. We mentioned before that phenotype, very generically speaking, is the result of genotype plus environment plus interaction of genes and their environment. Sex determination, sexual differentiation is no different. So there are species in which there is a strong environmental component to sex determination. So the embryo, as it is developing, it will take cues from the environment, telling it whether it should go towards the route of male sexual differentiation or female sexual differentiation. From there, it will develop the corresponding anatomies and make the corresponding gametes. Okay? So the take-home message is, despite this correlation with the presence of sex chromosomes, ultimately, sex determination is a genetic phenomenon. And as a genetic phenomenon, it is prone to variations of several kinds, and in some cases, strong environmental influence as well. Yeah?
The SRY gene is only in humans or all mammals?
All mammals. Cool. So that was sex determination, right? The connection between genetics and sex. When we say sex-linked genetics, we're not necessarily talking about that. What we're talking about is the inheritance of certain traits that seem to be strongly correlated or strongly influenced by sex, by biological sex, if you will, right? So sex determination is how we go from genes to sexual characteristics and dimorphisms. Sex-linked genetics is how certain genetics are linked to sex. So it all started with someone named Thomas Morgan, who over 100 years ago noticed something peculiar. He was working with fruit flies. He was sort of the founder of fruit fly research. And he noticed that if he crossed a pure breeding line of flies that had white eyes, these were mutant flies because normally fruit flies have red eyes. So he and his team had generated this line of flies that had white eyes instead. So if he crossed white-eyed males to red-eyed females, 100% of the F1 generation had red eyes. So knowing what you know about Mendelian genetics, if I asked you which one is dominant, having red eyes or white eyes, what would you say? Red eyes, right? And what would you expect from the F1 hybrid cross or monohybrid cross? In other words, what would you expect if we were to cross brothers and sisters in this F1 generation. With flies, unlike flowers, you cannot self-fertilize them, so you need a brother to mate with a sister. Make of that what you will. Let's not go crazy with you anthropomorphic analogies, but yeah, a brother and a sister can mate. What would you expect in the progeny?
Some red and some white.
Some red and some white. How many reds to whites? 3-to-1. I'm hearing some 3-to-1s, right? So you would expect a 3-to-1 phenotypic ratio in the F1 monohybrid cross. However, there were two strange things. First, when Morgan would conduct the reciprocal cross, so he would cross white-eyed females to red-eyed males, now the progeny, the F1 progeny, was not 100% red-eye. And that was unexpected, because if we had established that red dominates over white, then you would have expected that 100% of that F1 would have red eyes. Instead, all the females had red eyes, but the males, all of them had white eyes. And then another thing that was unexpected is that F1 monohybrid cross did generate a 3-to-1 ratio, but with a strong, actually an extreme sex bias, meaning that all of the females were red-eyed, 100% of them. Among the males, half of them had red eyes, half of them had white eyes. In the totality, there was a 3-to-1 ratio. But it was very peculiar that no females would have white eyes and only males would have white eyes. So Morgan concluded that the inheritance of this white eye allele or mutation showed a very strong sex bias. And he kind of established this idea of sex-linked genetics, meaning that now we need to start understanding these patterns of inheritance that are somehow linked to sex. It turns out that around the time, Sutton had proposed this chromosomal theory of inheritance that we briefly touched on before. This idea that when you look under a microscope, chromosomes look and behave like what Mendel had proposed for the units of inheritance, right? They came in pairs, they separated during meiosis, they recombined during fertilization. So Sutton had established this idea that chromosomes were like physical vessels for Mendel's units of inheritance. Morgan knew that females in flies had 2 X chromosomes and males had an X and a Y. In humans, the X is a really large chromosome and the Y is a tiny chromosome. In flies, size-wise, the X and the Y are not much different, but the Y is pericentric, whereas the X is acrocentric. So they look a little different, but they're about the same size. So Morgan started entertaining this idea that maybe the gene for eye color used these sex chromosomes as their vessel for getting transmitted to the next generation. So let's try and think about this together. Could the Y chromosome be the vessel for the eye color locus? Let's try loss of function. So let's say that you hypothesize that the Y chromosome is the one that carries the eye color gene, right? So if that is true, then you could try and find a Y chromosome with a mutant eye color gene, right? So if a fly had a Y chromosome with a mutant eye color, what would that fly look like? It would have the mutant type, right? And would it be a male fly or a female fly? A male fly, because it has the Y chromosome with a mutant eye color gene. What would females look like? What would be their eye color? Red? But I thought we said that the eye color gene is in the Y. That makes sense, right? If the eye color gene is in the Y chromosome, females should have no eye color because they have no Y chromosome. So right off the bat, through this exercise we did together, Morgan already knew that if the eye color gene lived in one of these sex chromosomes, it had to be on the X. Females don't have a Y chromosome, but they do have eye color. So the gene making the eyes red could not live in the Y chromosome. Otherwise, females, natural fly females, wouldn't have red eyes. So he starts entertaining the idea that the gene for eye color lives on the X, and that changes the way of approaching this genetic process completely. So instead of thinking, when I cross a white-eyed female to a red-eyed male, I'm crossing a homozygote mutant for this eye color gene, which he named lowercase w for the white mutation, white eyes, cross it to a homozygote male. Now he started thinking the female that is homozygote mutant will have one copy of the mutant gene in each of her X chromosomes, and the male will have only one copy of the wild-type gene for eye color in his only one X chromosome. The Y chromosome, we already established, does not carry this gene. So the Punnett square that he would have used to solve this cross would have looked something like this, where females would be providing X chromosomes with a mutant allele for eye color in them, and males would make half the sperm with an X chromosome with a wild-type allele and half the sperm with a Y chromosome, no allele because there is no locus for eye color in the Y, right? So using a classic Mendelian approach, you would have predicted from this cross between pure breeding flies with red and white eyes that 100% of the progeny would have been heterozygote for the mutation, and because the mutation is recessive, then those flies should have had red eyes. But using this approach, you can predict that of 100% of the progeny, half of them would be females, and they would have red eyes because those females inherited one X from mom with a mutant eye color gene, but then the other X from dad with a wild type eye color gene. Whereas their brothers, the other half of the flies, their brothers inherited Y from dad, but they inherited their only X chromosome from mom, and that one carries a mutant for the eye color gene. So because they are hemizygotes for this mutant allele, they have the mutant allele and nothing, they show the mutant phenotype, the white eyes. So the prediction was 100% of the females in the progeny would have red eyes, and 100% of the males would be wide-eyed. And that's exactly what he saw. Okay? So thinking about the inheritance of these particular trait as being linked to these sex chromosomes, and knowing that the sex chromosomes establish or determine biological sex in addition to eye color and many other traits, that's how Morgan could establish that the inheritance of eye coloration was sex-linked. So this slide here shows that you can work it out for the other cross. So if you remember, the first cross that we discussed was red-eyed females to white-eyed males. Now thinking about the genotypes as shown here, so the red-eyed females would be homozygote, wild type for the eye color gene, both of those copies on the X, whereas the white-eyed males would have one X chromosome with a white eye allele, and then the Y chromosome without anything in it. When you conduct this cross, the male is producing half the sperm with an X chromosome with a mutant allele and half the sperm with a Y. The pure breeding female is producing eggs that can carry only an X chromosome with the wild type allele. And when you solve, you're going to have 50% females with a heterozygote mutant for eye color. And because the mutation is recessive, these flies would have red eyes. And the males, the other 50% of the progeny, would have one X chromosome from mom with the wild-type allele for eye color and would therefore have red eyes. Now, so far so good. If you remember, for this cross, things broke down when Morgan carried out those F1 hybrid crosses, because there was that 3:1 phenotypic ratio, but it had a very strong sex bias. So when you start thinking about that, F1 hybrid cross, so this is the F1 hybrid cross, in terms of the females being heterozygous, so now they produce two kinds of eggs. Half the eggs will have the X with the wild-type allele, and half the eggs will have the X with the mutant allele, whereas the males are still producing, I'm going to change color here, are still producing half the sperm with an X with a wild type and half the sperm with a Y. You solve this Punnett square, and then you notice that there is 50% of the flies are female, but all of them have red eyes, because half of them are going to be homozygote wild type, and the other half will be heterozygote. And since the mutation is recessive, they're going to show the wild type phenotype, the red eyes. Whereas the males, again, 50% of the progeny, but half of the males inherited an X with a wild type from mom, but the other half inherited an X with a mutant. And in those males, that mutant allele is hemizygote, and it expresses phenotypically as white-eyed males. Okay? Did that make sense? So we're not going to really talk about sex-linked genetics in flies or in any of those other organisms that we discussed. From now on, it's mostly going to be about sex-linked genetics in humans. But the process is the same. It's thinking about these loci that live in sex chromosomes and therefore are going to be somehow tied to what those sex chromosomes do for an individual, which is sex determination as well. Okay, so, and hence the strong bias with the biological sex of the individual. So, switching to humans. I'm not going to ask, but statistically, there may be one or more individuals in this classroom right now that may not see the number inside of that circle. And that is because there is a form of color blindness that is very widespread in the human population that is sex-linked. So this particular form of color blindness is based on a locus that is located in the X chromosome. And what happens is just like we described for flies, you can go check it out later, but because this allele is on the X, for a woman to experience this form of color blindness, she has to be homozygote recessive for this. So both her X chromosomes need to carry the mutant allele, or the allele for this form of color blindness. For males, all they need is one copy of this mutant allele because they're hemizygotes. And if you were to look at a normal color vision female here that has progeny with a colorblind male, you can work out that all of the progeny will be normal color vision. Whereas if you were to have a colorblind female with a normal color vision male, now half the progeny, the girls, the daughters, will have a normal color vision because they get the X from dad with the wild type allele for this gene, whereas the boys, the sons, are colorblind because they get their only X chromosome from mom. So just like we saw with the flies, you could almost imagine the white eyes being this particular form of color blindness, and it would track perfectly. Okay?
Does that mean it's more common for males to be colorblind?
We'll talk about that. So it is more common for males to experience this kind of color blindness. The way that you phrase the question, is it more common for males to be colorblind? There's other forms of color blindness, and if those live in autosomes, then there is no way to tell. If there is any color blindness that is X-linked, but is dominant, then all of a sudden it's more common in females. if there were any condition, let's say color blindness, to keep with this argument, a color blindness that is linked to the Y chromosome, then only males can experience that form of color blindness. Does that make sense? Yeah? But we'll discuss these different patterns of inheritance today. So that brings us to pedigrees, and we needed to sort of discuss sex-linked genetics as a concept so that we can understand how we evaluate pedigrees. Some of you may have seen this before, but pedigrees is just a fancy term for a family tree, right? And in pedigrees, there is certain notation that you need to be familiar with, also indicated with blue arrows here on the right. You need to know that males are squares, females are circles. Whenever we see a diamond, that's because the sex is unknown or unspecified. That may sound a little silly, but when you're constructing a family history and you're going several generations back, some people might say, I think there was a brother or a sister, I can't remember, but this person had the condition. So whenever a medical geneticist can't specify the sex of an individual, they use a diamond. Empty shapes mean that that is an unaffected person. Filled shapes, it means that the person is presenting a condition or disease. A shape with a dot inside means that those persons are carriers of recessive alleles for a condition that is recessive. So it's simply saying they're heterozygotes for those alleles. If there are many individuals in a family tree that are siblings, then often what medical geneticists will do is just collapse them all into one shape and indicate how many of those there were with a number. So if there's a family of 4 sons and 5 daughters, there'll be a square with a 4 inside and a circle with a 5 in it. Any deceased person is a shape crossed. Families are indicated such that the parents are linked by a line, and then you see these sort of branches that branch out to their children. And last but not least, in a family tree, whenever you see 2 lines connecting individuals, it means that, yes, those individuals procreated, but also that they were connected by blood, right? So it's what we call consanguinity, and it's to indicate what we, in more late terms, we would call inbreeding. Right? They don't necessarily have to be brother and sister. It could be cousins as well, but there is some sort of family connection between them. Okay? So now let's go through these different types of inheritance that are associated with different kinds of conditions. So the easiest one, perhaps, is autosomal dominance. This means alleles that live in an autosome, not in one of the sex chromosomes, and if the individual has the allele, they express the phenotype, they express the condition, because this allele is dominant. So only one copy of that allele will express phenotypically. There are some features to autosomal dominant conditions. First of all, autosomal dominance do not skip generations, meaning that, we'll discuss in a second what that means. And because they're autosomal, there is no sex bias. So if you look at a large family tree, you should see that roughly, the condition affects females and males in the family equally. Okay? Let's talk about skipping generations. So no generation skipping doesn't mean that 100% of the progeny inherits the trait. That's a common misconception. So let's look at this couple here. Male 6 has children with female 7, right? And you can, 6, 7. I swear I didn't plan this. I see these things and I get the, again, you missed it again. 6. Then I throw myself off. 6 and 7 have these 3 kids, right? 2 daughters and a son. And notice how not all of them inherit the condition. So no generation skipping doesn't mean that every child inherits the condition. It means something different. No generation skipping has to do with, I have it here. So let's work through this example. What do you think? Is this condition, whatever the condition is, doesn't matter. Is this condition skipping generations? When we're thinking about skipping generations, we shouldn't necessarily look at the generations by themselves. So the condition is present in every generation. You look at these grandparents, grandparents, parents, and children. In all of the generations, the condition was present, which may have led some of you to say it didn't skip generations. But when we're trying to evaluate if a condition skips generations or not, we need to look at lineages. We need to look at parents and their children and their grandchildren. So here are two examples in which the condition shows up from parents, the line is a little screwed there, but from parents that didn't have it, right? So whenever you're trying to evaluate if a condition is skipping generations or not, and that will matter when you're trying to determine modes of inheritance, if you're trying to answer the question, is this condition skipping generations or not, home in on any individual with the condition and check their parents. If you find any case in which an individual has the condition but neither of their parents did, then it means that the condition skipped their parents' generation. And if you keep going back, you might find even that grandparents didn't have the condition either, and you may have to go 2 or 3 generations back to see it again. So generation skipping, we may be tempted to go forward, go backwards, home in on the latest generation you can and start going up through the parents and see if there is any cases in which the parents don't have it. And then you've established that that condition did skip generations. You guys gonna remember that? Yeah, cool. All right, so we just talked about autosomal dominance, and the reason why they don't skip generations is if the allele is passed on, then it will show up, right? And vice versa. If a child has the condition, either or both of the parents had to have it too, and they had to show it because it was dominant. It didn't matter if they had 1 or 2 copies. Even with 1 copy, either parent would have had to show it. So autosomal dominant don't skip generations. What about X-linked dominance? Well, X-linked dominance don't skip generations either, right? But they do affect males and females slightly differently. So females with an X-dominant condition behave as if they had an autosomal dominant. Why? They have 2 X's, so for females, an X-linked condition is not much different from an autosomal dominant condition. All of their gametes will have an X in them, and whichever X had the dominant will pass it on to the next generation. So for females, it's indistinguishable. But X-dominant affected parents do not pass the condition to sons. And that should say fathers there, not parents, fathers. So X-dominant affected fathers do not pass the condition to sons. So that is the hint that we look for to distinguish between an autosomal dominant and an X-dominant. The difference between an autosomal dominant and an X-dominant is males will pass autosomal dominance to their sons equally as their daughters. But when it comes to X-linked X-linked dominance, they cannot pass it to the sons. What is it that they're giving their sons? The Y chromosome, right? So their X chromosome with the dominant allele in it is not passing down to sons, it's only passing down to daughters. So if you're trying to establish if a dominant condition, you've already established that there is no generation skipping, a good sign that that is a dominant condition. If you're trying to distinguish between autosomal dominant and X-linked dominant, look to see what happens to the fathers. If the fathers are consistently passing it to girls, to their daughters, and not sons, it's a good sign that it's an X-linked dominant. X-linked dominant conditions are more prevalent in females, so this is in a way going back to your question before, and that is because affected females will pass it with equal probability to daughters and sons, but if the affected parent is the dad, only daughters get it. So if we assume for a second that there is equal number of couples in which the female is affected and couples in which the male is affected, In both cases, the daughter get it, but only in one case will the sons get it. So from a population perspective, X-linked dominants are actually more prevalent in females. Okay. Let's turn the page and let's start talking about autosomal recessives. Autosomal recessive traits usually appear equally in males and females. That is because there is no linkage to sex chromosomes. So these things travel in autosomes and they get passed on to daughters and sons equally. They tend to skip generations because they're recessive conditions. So there is that necessity that both parents contribute a recessive allele to a child for that child to manifest physically the condition phenotypically. And autosomal recessives are more likely to appear among progeny of related parents. So again, we go back to this idea of consanguinity. This is common. This was common historically in communities in which there was a lot of inbreeding within families. Nowadays, you can see it more commonly, let's say, among in a farm among cattle or dogs or domesticated animals in which there can be a lot of inbreeding as well, a little less in humans. One thing that I don't like about this slide and that you see a lot in text is this idea that autosomal recessives are more common when there is consanguinity. That is true, but I don't want you guys to walk away with the idea that you will only see it when there is inbreeding. That's not true, because there are many recessive alleles that are a lot more prevalent in the population, and you can have two individuals that are completely unrelated having children and then happen to be both carriers of these recessive alleles, so they may not manifest the condition, but they do carry one copy of these alleles. They pass it on to their children, and the condition appears, right? So this idea that these types of conditions are more common when there is consanguinity doesn't mean that there has to be consanguinity for them to appear. Yeah? We good? In a way, this sort of rephrases what I just said. With rare autosomal recessives, unaffected outsiders, what we mean by that is individuals that are not affected by the condition and come from outside of the family, usually don't carry the allele. And this is for rare autosomal recessives. These are recessive alleles that are rare in the population. So that's why autosomal recessives tend to skip generations. And it is actually rare for those autosomal recessives to reunite in progeny, unless, of course, not progeny. It is rare for them to reunite in future generations unless there is some sort of consanguinity. So with rare autosomal recessives, once it skipped a generation, it will never come back. For that family tree, at least, or for that family lineage. It's rare that they come back. Unless via consanguinity breeding. Now, on the other hand, and notice how I now denoted those dots in each of these children from that original couple, because even though none of them expresses the condition, none of them inherited the recessive allele from mom, they all carry the recessive from dad. Why am I saying that they all carry the recessive from dad? Why couldn't it be that some of them inherited the recessive from dad, but some of them didn't?
Because the dad is affected.
Exactly. And if the dad is affected, he must be
Two sets of
recessives. Exactly. So the dad must be homozygote for the recessive, so he's passing the recessive allele to all his children. Even though none of them may manifest the condition because they didn't get a recessive from mom, they all carry the recessive allele. Now, what would happen if that recessive allele is a lot more common in the population? So now you have an affected individual and an unaffected outsider. If the allele is common in the human population, she, in this case, may carry. She may be a carrier. Right? And in that case, the condition may easily appear among their children. Okay? We're good? So all of these about autosomal recessives. They tend to skip generations. If they're rare, if they skip a generation, they never come back. There is no sex bias. And last but not least, the fact that they may skip generations doesn't necessarily mean that when you have an unaffected individual procreating with an affected one, they have to be cousins or rather than siblings or anything for them to pass it on to progeny. If the allele is prevalent enough in the population, they can show up just by chance. Yeah? Last but not least, the X-linked recessive. So in a way, we talked about this already with that color blindness example and with the flies. The idea is here that they do have a strong sex bias. So X-linked recessives affect males more often than females. And that is because a female needs 2 copies of a recessive allele in her X's to manifest the condition. Males need only 1. So it's a lot easier for a male to inherit 1 X chromosome with that 1 recessive copy, and he's already manifesting the condition associated with that allele, whereas females require 2, and there may be a lot of female carriers out there, but they're not showing the condition. Like X-linked dominance, affected males do not pass the condition to their sons. So one telltale that something may be X-linked is when you see that the dads are not passing it to their sons. And the counterpoint is that all daughters of an affected male are at least carriers. All daughters of an affected male will inherit that 1 X chromosome from dad with the recessive allele. If mom passes an X with a recessive allele as well, they show the condition. If mom passes an X chromosome without a recessive allele in it, well, at least they're carriers. Did that make sense? It's a lot to take in, but you'll have some time to sit down with these trees and these kind of sets of rules. But I wanted to sort of exercise with you a little bit. So imagine that you have these 2 trees, right? These 2 pedigrees. What type of condition do you think are consistent with these pedigrees? I'll give you a minute or 2, talk with friends, make friends, talk to your neighbors. So what are we thinking? I know that you had your hand up before.
I was thinking one of them was an X-linked dominant.
Which one, the left or the right?
Probably the, X-linked recessive on the right side. Or left side, I mean.
The left side. So X-linked recessive?
Yeah.
Okay. Mom has the condition, passes it only to sons, and all of them. So yes, that is consistent with an X-linked recessive. If it were an autosomal recessive, then you would see some daughters with the condition as well, right? Because some of the daughters, and actually if it were an autosomal recessive, you would see some of the sons not having it too if that is not a carrier, right? So yeah, this is very aligns very strongly with X-linked recessive. What about the right hand side pedigree? Same. X-linked recessive. So that is passing the X's to his sons.
It's all the Y chromosome.
Y chromosome, right? We didn't even talk about that, but now that dad is passing it only to sons, what is the one thing that dads pass only to their sons? The Y chromosome. So this may be Y-linked. Again, we absolutely uber-duper confirmed it? No. There could be other scenarios that might give rise to these pedigrees, but they would be rather unlikely. So for instance, the right-hand side pedigree, that could carry an autosomal dominant, for instance, and he's just, luck had it that he passed the dominant to the sons and not the daughters. What are the chances of that? We can calculate it. What is the probability that a daughter inherits it times the probability that the second daughter doesn't inherit times the probability, and so forth. It will be an extremely small number, I promise, right? So this is a little more consistent with a Y-link. Have we absolutely confirmed it? No. What about this, pedigrees? Let me tell you right now, it gets a little more complicated. We have only one generation. We have no idea to determine. So this, while there was no generation skipping right here, there could have been generation skipping if we looked at other sides of the family. There doesn't seem to be sex bias, right?
Autosomal dominant?
It could be autosomal dominant. It could be autosomal recessive if it is a common recessive in the population. So what the point of these two pedigrees is that it gets a little trickier when you get this type of distribution of a condition within a family. And it gets even trickier if, forget those extra brothers and sisters, if we're talking about couples that probably had a couple of children only. Right? So if I showed you any of these two really tiny, small, one family core pedigree, and I asked you, what is the mode of inheritance there? I don't know, right? What is the point that I'm trying to make here? The point that I'm trying to make here is that in human quote-unquote crosses, let's say procreation, there's usually not enough information to strongly support one mode of inheritance over others, over another. So it could always be consistent with something else. The one thing that we can do, though, is in certain circumstances use a process of elimination to say it could be a number of things, but it cannot be this. For instance, you see a father passing it to a son and a daughter, so now you know it's not Y-linked, right? And so forth. You see generation skipping, so now you know it's not dominant. So there are certain things that we can rule out by process of elimination. So we can often home in step by step on a mode of inheritance by inspecting different areas of a pedigree and using different areas of a pedigree to rule out certain ones or certain modes of inheritance. So one area of the tree might say, oh, this says that it's not Y-linked. Oh, this says that it's skipping generations, so it's not dominant. Oh, I see that a father is passing it to a son here, so it cannot be X-linked, and so forth. Yeah. There is, in Canvas, one of the homework quizzes called Pedigrees 2 deals with precisely with this issue of how we approach pedigrees as a stepwise process of elimination process by which we start ruling out certain things until we home in on a given mode of inheritance. Okay, so make sure to check it out. And the last thing that I wanted to mention before we move on to the quiz is that all this business of human crosses or say human procreation, we sometimes don't know the relevant genotype of the parents. If you tell me a couple comes and they tell me that for a given locus, he's homozygote wild type, she's heterozygote for a recessive allele, then we can calculate what are the probability that children carry the allele or don't and never pass it on to grandchildren, etc. But often we don't know the relevant genotype of the parents. And why don't we know? Well, there's two reasons. One is, even though genetic sequencing is getting cheaper and cheaper by the day, it's not that cheap still. So for many people, it's kind of prohibitively expensive to go to the doctor and the doctor says, well, we can run a genomic sequencing and it's only $10,000. There's still a price tag, okay? So that's one reason. And the other one is, and this is sort of more universal, there are many conditions for which we have a very good idea of how they are inherited. So we can say with certain certainty that those conditions, based on what we know about family trees throughout history, that a certain condition is autosomal dominant or autosomal recessive or this or that. but we don't know exactly where the gene is. So even if we could sequence the genome of individuals, we wouldn't know where to look, right? So there's many cases in which you would like to predict what is the chance that a given child will carry a condition, allele, or actually suffer a condition, but without knowing the genotypes of the parents is kind of tricky. But have you guys heard of something called the Hardy-Weinberg equilibrium? Are you getting a little PTSD from ecology classes? Yeah? It turns out that we can use the Hardy-Weinberg equilibrium to work around this problem of not knowing what the genotype of parents is in order to make fairly well-informed decisions or predictions as to what the children might be like. So we can use Hardy-Weinberg equilibrium to make these predictions. There is a page in Canvas at the end of this week's module called From Mendel to Population Genetics, and I strongly encourage you to check it out. There may be 1 or 2 questions associated with that in exams, and it covers how we go from simple Mendel genetics or Mendelian genetics to how we derive the Hardy-Weinberg equilibrium. Okay?